So hi everyone, this is our experiment about photosynthesis. Very fun experiment, very beautiful experiment. Once you understand the whole process of photosynthesis and what happens in photosynthesis, you will learn by now that we need intakes and outcome. The intakes are sunlight, carbon dioxide, and water, and the outcomes are oxygen and sugar. Now, I need you to learn that photosynthesis happens in the leaves. Why in the leaves in particular? The leaves of the plants, because chloroplast is numerous in the leaves, and photosynthesis takes place in the chloroplast. On the leaves itself, on top of the leaves and bottom part of the leaves, you will find an opening. Of course, this opening you won't be able to see it with your naked eye. We need to use the microscope for that to view. These openings are responsible for the gas exchange, where the where the carbon dioxide enters and the oxygen comes out to us. Okay? Now, the plant will sense that sunlight is there, carbon dioxide is there, water is there, and will form the photosynthesis process. We will do a very simple experiment. We will use two leaves. One was kept away from the sun and one was kept facing the sun, to look whether the photosynthesis will take place, basically the gas exchange. We noticed that after small experiment, and we view the stomata under the microscope, is that when the sunlight is there, the stomata is opened. Look at the stomata. This is the opening, and this is the closed stomata when we keep it away from the sun. We're just, like playing with the stomata, okay? So that simple experiment, I will show it to you in a minute, but I need you to look at the opening. It looks like an eye, open eye and closed eye, and this is the lids are really closed, okay? Take a very good careful while I'm performing the experiment. Learn the materials, learn the experiment, because you will have to write a lab report. Your purpose today is learning about the importance of stomata. That's it in photosynthesis, okay? Now, as you can see, this is what we need for our experiments. Yes, this is nail polish, clear nail polish. I need leaves. I use this as facing the sun, and we can use it with the one non-facing the sun. I have the result for both. We need scotch tape, and we need microscopic slide. Now, how we would perform this experiment? Very simple. I will use the bottom side of the leaf. Now, the reason I'm using the bottom side than the upper side is the chloroplast, or the stomata is numerous in this area. It's much more than the upper part. So I'll just use this. Very simple. I will take some of the nail polish. I will apply it on a small area. Now, the reason I'm doing this, see, this is the area where I applied the nail polish. The reason I'm doing this is that once the nail polish is dried, and I will use the scotch tape to remove the nail polish. I'm not removing the plant or the stomata or anything. I'm just removing the dried nail polish. It will take a print of the shape of stomata. So if it's open, you will be able to view it open. If it's closed, you will be able to view it closed. So I'm printing the stomata. I'm not removing anything. I will wait for that to dry like for 5 to 7 minutes, and then I will apply my Scotch tape. I won't apply it directly. I will wait until this small spot is really dried up. So once the nail polish is dried, in this point of a step, I can add 1 drop of iodine for color, or I cannot use it. This is an optional to use the iodine as a color. I will not use it because it is really obvious under the microscope. So I'm just letting you know this information. So I will grab the Scotch tape, I will apply it, and make sure that you don't get your fingerprints on this area because you will be viewing it under the microscope. Press really good. Now, before I will apply it on the slide, I need you to, just a second. I will remove the lights for you to view it. Can you see? This is the dried nail polish. So the print, the shape of the stomata will be printed on the dried nail polish. I will apply it on the microscopic slide. And it's ready to be viewed under the microscope. It will be similar to these one I already have done with the previous years ago. Okay, so very simple. You will view it under the microscope. You have the result in your PowerPoints. You can use them in your lab report. You have the opened and closed stomata. Okay.